morning. I'm Dr. Niti Maheshwari, Assistant Professor, Applied Science and Humanities Department, Ajay Kumar Garg Engineering College, Ghaziabad. In the last lecture, I was taking UV visible spectroscopy, and during the last slide, I was taking the term auxochrome. I've already taken chromophores, which are color developing or color showing groups which are present in the organic compounds. Now coming to the next term, that is the auxochrome. Now these auxochromes, they are not color developing groups, they are color enhancing groups. Now when these auxochrome, they get attached to the chromophore, then they changes the intensity of the chromophore, they intensify the intensity of the chromophore or the color produced by the chromophore. It is due to their ability to extend conjugation. Certain groups which act as auxochromes, they are NH2, OH, OR, NHR, SH. There are various groups which can act as auxochromes. Now, here I am showing some examples, some groups which can act as chromophore and some which can act as auxochromes. Now in the first case, benzene, it contains unsaturation, pi electrons are present, so they can undergo pi to pi star transition. It acts as a chromophore. But when I add group like OH or NH2 to this benzene, then these OH or NH2, they can increase the conjugation because they contain lone pair of electrons. So these electrons can extend the conjugation of benzene, and hence as a result the value of lambda max increases. So a new chromophore is formed by the combination of a chromophore and auxochrome, and they intensify the color. As a result, the value of lambda max, it increases. Now next, coming to the different types of absorption and intensity shifts. Now there are 4 different types of shifts which we can see in UV visible spectroscopy. First is the bathochromic shift or the red shift, then hypsochromic shift or the blue shift, then there is hypochromic effect, and the last one is the hypochromic effect. Now in this diagram, I am showing all the 4 different types of shifts which can take place. This graph is between absorbance and wavelength. So there are 2 wavelength shifts and 2 absorption shifts. Now this is the graph of any compound which is showing the absorption positions in the UV visible region. For any compound that we want to determine, we study its graph in the entire UV visible range from 200 to 800 nanometers, and there is a position at which it shows maximum absorption. Maximum absorption of light takes place, and that is known as absorption maxima position, and that wavelength at where the absorption maxima is taking place, it is known as lambda max or the wavelength maxima position. Now if by any effect the value of this wavelength, if it changes, then it can show the bathochromic shift or the red shift. It is towards the longer wavelength side, towards the red end of the spectrum. Vibgyor, so towards red end, that is the longer wavelength side, so it is known as the red shift. Then if somehow the value of this lambda max shifts towards lower wavelength or the higher frequency side, then it is termed as hypsochromic shift or the blue shift, that is towards the blue end of the spectrum. Now, for example, if I take CO, in the case of CO, the N to pi star transition, it shows a red shift if I decrease the polarity of the solvent. Now, what happens if this compound is taken in a polar solvent? Then these N electrons, the lone pair of electrons, they tend to form a bond with the solvent. They can form a hydrogen bond. Now, these non-bonding electrons, they get bonded with the polar solvent. So, to excite these non-bonding electrons, you require higher energy. But if I decrease the polarity, then I am breaking the bond which this lone pair of electron makes with the solvent. When I break the bond, then these electrons, they become free, and therefore it is easy to excite these lone pair of electrons to the higher orbital side. Therefore, a red shift is seen. Lowering of energy increases the wavelength, therefore red shift. Coming to the blue shift, which is the hypsochromic effect, if I break the conjugation, then this type of effect is seen. For example, this molecule is aniline. Now, in normal conditions, this lone pair is involved in conjugation, but if I change my solution to acidic, then this aniline, it changes to, in acidic medium, it forms NH3 plus. Now, these lone pair are no longer available for conjugation. Hence, I am breaking the conjugation. Therefore, this type of absorption shifts towards the shorter wavelength side or the higher frequency side. Then, there are two absorption shifts. If my absorption intensity gets increased, it is known as the hyperchromic effect. Generally, the presence of auxochromes increases the intensity. If I add auxochromes, then this effect is seen. Next, hypochromic effect. It is the effect by which the position of the absorption maximum decreases. If this position decreases towards the lower side, it moves towards the lower side, then it is hypsochromic shift. It is generally seen in those compounds where their symmetry gets distorted. For example, in the case of biphenyl, biphenyl is a highly symmetrical structure, but when I change this biphenyl to 2-methyl biphenyl, then I am distorting the geometry of biphenyl, and hence in that case hypsochromic shift or effect is seen. So, overall there are 4 different types of shifts: red shift, blue shift, increase in wavelength, decrease in wavelength, increase in absorption maxima, that is the hyperchromic effect, and decrease in absorption maxima, that is the hypochromic effect. Now, coming first to the bathochromic effect or the red shift. Now, when absorption maxima of any compound shifts towards longer wavelength, then it is known as bathochromic shift or the red shift, as I have already discussed. Now, this effect is due to the presence of auxochrome or by the change of solvent. So, both of these effects can cause the bathochromic shift. And exochrome groups like OH, OCH3, they causes absorption of compound at longer wavelength. Next is hypsochromic shift, that is the shifting of the wavelength towards the shorter position of the lambda max towards the shorter wavelength side or the blue shift, then it is maybe produced by the removal of the conjugation, as I have already discussed, and by the change of medium. Now, the solvent effect is also seen in the case of hypsochromic effect. Next is hyperchromic effect, that is the absorption maxima position increases. Now, this is usually affected by the introduction of auxochromes. If I add an auxochrome, then this effect can easily be seen. Again, the hypochromic effect, by which the position of the absorption maxima decreases, that is the epsilon max decreases. Now, it is caused by the groups which distorts the geometry of the molecule. Now, coming to the instrumentation in UV visible spectroscopy. Now, the instrument which is used in the absorption of UV or visible light, they are made up of the following components. First is the light source, then there is a filter or a monochromata, so that single wavelength light passes through it. The sample containers or the sample cells, generally these are the cuvettes, and lastly that is the detector which detects the radiations or the absorptions. Now, this I am showing the diagram of UV visible spectrophotometer. Spectrophotometer is a device used for the studying of the absorption of radiations. This is the light source, light of many wavelengths is passing through it. A monochromata or a grater is used, which allows only the passage of a single wavelength light. This is the sample solution which is present in the cuvette. This is the intensity of light before absorption. This is the intensity of light of transmitted radiation. Then the detector, which is generally a photocell, and then finally the digital display or the meter, which shows the how much absorption or transmittance has taken place. Now, coming to the radiation source. The different radiation sources which can use, or the radiation source should be such that it studies the entire UV visible spectrum. So, different types of lamps are there which cover the entire region. They are hydrogen discharge lamp, tungsten lamp, mercury lamp, and they should require, or they should meet the certain requirements, like they should be stable and they should be able to provide continuous radiation. Now, the cuvettes in which we take the sample, it is generally made up of quartz cuvette. If work has to be done in the ultraviolet region, and the cuvettes, they may be rectangular or they may be cylindrical. Generally rectangular cuvettes are taken. Now, these rectangular cuvettes, they are transparent on the two sides and translucent on the two sides. Then sample containers or the sample cells, they are the cuvettes again. The test sample is taken in the cuvette which is to be analyzed. It is placed between the detector and the monochromata, and the walls of the cell in which the test sample is placed, it should be transparent to the radiation used, and light should pass straight. Therefore, the other two sides are translucent in nature. Now, coming to the detector which detects or which is used to measure the absorption of the analyte sample by the intensity of the transmitted radiation, how much light has been absorbed. It is calculated by how much light is getting transmitted. So, there are three types of photosensitive devices which are used. It can be photovoltaic cell, then phototubes or photomultiplier cells which can be used. Now, coming to the applications of UV visible spectroscopy, where we can use this spectroscopy. So, the different applications of UV visible spectroscopy, they are: first, it is used for the characterization of aromatic compounds and conjugated olefins. So, aromatic compounds and conjugated olefins, because they contain pi electrons, by Woodward-Fieser rule we can calculate the unsaturation which is present in the organic molecules. Then it is used for the detection of impurities. If impurities are present, then the absorption position is not going to be perfect, it is going to be broad. Then detection of isomers by the UV visible spectroscopy, whether the isomers they contain double bonds, those isomers which contain double bonds in conjugation or which have double bonds other sides at different intervals. Determination of molecular weight can be done by Beer-Lambert's law. We can study the kinetics of the chemical reaction, how much reactants had been converted into the products. Then it is used in quantitative analysis of pharmaceutical substances and drugs. Now, in the last class I had said those systems which contain double bonds in conjugation, they show absorption at longer wavelength. That is, question arises, why conjugated systems have higher value of lambda max? If I take a non-conjugated system, then it contains a pi orbital and a pi star orbital, non-conjugated system. Pi orbital which is the bonding and pi star which is the anti-bonding. So, there is a specific distance energy difference between the pi and the pi star orbital. But when I see the same case in the conjugated systems, then here there are two pi orbitals and two pi star orbitals which are being formed. Now, in the first case, that is psi 1, this psi 1, as you can see, same colored lobes are present on all the sides, red end on one side, black one on the other side. So, same lobe, that means all are plus on one side, minus on the other side. Now, these pi and pi, it results, psi 1 results from the combination of pi plus pi. The pi orbitals of both the double bonds, it results in the formation of psi 1, and this is in phase combination. Out of phase, but in the bonding it contains one node. This is plus plus, minus minus. So, plus plus overlapping, minus minus, minus minus and plus plus. So, there is one node which is present in between and it results in the formation of psi 2. And this psi 2 is the highest occupied molecular orbital, and this psi 2, it is formed by the combination of pi minus pi. Now, third, which is psi 3, again you can see that minus plus, plus minus, plus minus, minus plus. So, as a result there are two nodes which are present in the system. First node is here, this is the second node. Two nodes are present and it results in the formation of psi 3, and it is formed by the combination of pi star plus pi star, and it is also the lowest unoccupied molecular orbital. And the last one, here you can see minus plus minus plus. As a result, there are three nodes in between, and it is formed by the combination of pi star minus pi star. So, when transition takes place in the case of conjugated dienes, it takes place from the highest occupied molecular orbital to the lowest unoccupied molecular orbital. So, the bonding energy gets elevated. You can see the psi 2 is at a higher energy level in comparison to the psi 1, and psi 3 is at the lower energy level in comparison to the psi 4. So, the transition of electron takes place between this level, and the energy difference between psi 2 and psi 3, it is lesser in comparison to the normal transition which is taking place between from pi to pi star. So, the energy difference of this psi 2 and psi 3 is lesser than pi to pi star transition. As a result, when the energy difference is less, therefore, this transition will take place at a higher value of wavelength. So, they have higher value of lambda max. So, if two examples are given, then the one containing longer conjugation or the higher value of conjugation will have a higher value of lambda max, and the other compound in which the conjugation is breaking in between, that will have a lower value of lambda max, and it will show the blue shift, and the system which have the higher value of lambda max will absorb towards the longer wavelength or the shorter frequency or at lower energy. And when the systems are not in conjugation, they are going to absorb light of higher frequency, that is of higher energy. That is why the conjugated systems, they absorb at higher value of lambda max. Now, next question comes, what is the effect of polar solvents on N to pi star and pi to pi star transitions? I have taken one example of CO. Now, if I take a polar solvent, CO bond is polar, carbon is delta positive and oxygen is delta negative. So, if I take a polar solvent, then suppose I have taken this polar solvent, then since oxygen already has lone pair and it is also pulling the electrons for the carbon, then these lone pair of electrons, they are going to form, or this hydrogen will form, a bond will be formed between oxygen and hydrogen. So, these lone pair, they tend to form a bond. So, first the lone pair were free to but now these lone pair are bonded. Since these lone pair get bonded, therefore, they require higher energy. If I take a polar solvent, then N to pi star will move towards shorter wavelength or the higher frequency side, because the lone pair of electrons, they get bonded with the solvent molecules, and once the electrons they get bonded, you have to break the bond to excite the lone pair of electrons. Therefore, the process becomes more energetic than when it was in the non-polar solvent. The second case, coming to the pi to pi star transition. Now, this CO bond, these are the pi electrons here. Here are the pi electrons, this is the pi bond. So, pi electrons, when I take a polar solvent, then the polar solvent tends to form a bond. When the polar solvent tends to form a bond, then this bond, it gets elongated. When these polar, because it is positive and negative, the polar solvent will also have positive and the negative ends. Therefore, they tend to form a bond, or they get attracted towards the opposite signs. There are electrostatic forces of attraction between the solvent and our sample. Therefore, this bond, it gets elongated. Now, this pi bond, which was stable before, the polar solvent is increasing its energy or decreasing its stability. When it gets increase in energy, that means we require lesser energy now to excite these pi electrons to move from the highest occupied to the lowest unoccupied orbital. So, when they move, this pi to pi star transition, it gets moved towards the longer wavelength side. So, in the polar solvents, our n to pi star, it moves towards the shorter wavelength, but pi to pi star, because the energy gets elevated, therefore, it tends to move towards the longer wavelength side. So, in polar solvent, n electrons require more energy than before, and the pi electrons require lesser energy than if polar solvent was not present. Thank you.